everybody. Welcome back to our final committee breakout session. This is advancements in genomic and genetic prediction. And our chair for this committee is Dr. Mark Thallman. Mark is a research geneticist at the U.S. Meat Animal Research Center in Clay Center, Nebraska. And Mark's been a longtime chair of this particular committee. And we're thankful for his dedicated service over time. Also a past BIF Pioneer Award winner, if I remember correctly. So Mark's made great contributions to both this committee and genetic improvement here in the U.S. Mark, I'm going to give you remote control, and you should be able to advance slides. I'll get to the first one here, hopefully. Maybe. Okay, yep. So here's Mark. It should be good to go. Whenever you're ready, I think you just need to click screen here, and it should revert control back over to you. So.
Okay. Thank you, Bob, and thanks for all you've done in getting this meeting coordinated. That's been a monumental task. So I want to remind everybody before we go further that the recordings and slides of the presentations are accessible on the schedule page for the conference. And so would encourage you to go back and look at anything that you've missed or that you need to get more details on. We're going to have two presentations in this committee session. The first one will be on low-pass sequence data and genetic evaluation, and the authors are Drs. Larry Kuehn, Warren Snelling, Mark Thallman, and Matt Spangler. And following that, we will have increasing accuracy of genomic predictions from SNP chips to sequence data by Dr. Daniela Lourenco. And before we do that, we want to take a minute to thank our sponsor for this session, which is Rabo AgriFinance. Our sponsors are very important for the success of the BIF convention, and particularly this year, since we don't have any registration fees, we're offering the conference at no charge to everyone who wants to watch. And we're happy to be able to do that, and very pleased to be able to do that because of the generosity of our sponsors. And so anytime that you have an opportunity to thank them, please do so. So our first presentation, as I mentioned, is low-pass sequence data and genetic evaluation. And the authors are, again, Dr. Larry Kuehn, Warren Snelling, and Mark Thallman of the U.S. Meat Animal Research Center, and I'm going to go through our bios kind of together. Larry Kuehn is from Nebraska and received a BS in Animal Science from the University of Nebraska, and MS. Did I do that, Bob, or did you?
That was
okay,
me, sorry.
Keep going.
Yeah, keep going.
So Larry received a BS in Animal Science from the University of Nebraska, an MS in Animal Breeding from Colorado State, and a PhD in Animal Breeding from Virginia Tech. Warren's from Kansas and received a BS in Animal Science from Kansas State University, and an MS and PhD in Animal Breeding from Colorado State University. And I'm from Texas and received a BS in Animal Science and MS in Animal Breeding, and a PhD in genetics from Texas A&M University. All three of us are beef cattle geneticists at the U.S. Meat Animal Research Center in Clay Center, Nebraska. Larry is the research leader of our unit. Together, the three of us co-lead the germplasm evaluation project at MARC. This project consists of about 3,600 cows representing highly influential AI sires of 18 breeds and measures a broad spectrum of economically important traits on the progeny. The GP population is used for estimation of breed differences across breed EPD adjustment factors, breed-specific heterosis, genetic correlations, and associations between genomic variation and economically important traits. Each of the three of us works on various approaches to genomic prediction. And our co-conspirator in this is Matt Spangler. Matt's not going to present in this presentation, but he contributed a lot to it. And Matt grew up in Kansas and received a BS from Kansas State University, an MS from Iowa State University, and a PhD from the University of Georgia, and is currently a professor and extension beef genetics specialist at the University of Nebraska. He works as part of a team with colleagues at UNL and USMARC to improve genomic selection tools and methods, and currently leads a USDA-funded project to develop web-based decision support tools for genetic selection. And he gave a great presentation on that in the Selection Decisions Committee yesterday, and if you missed it, I'd encourage you to go back and take a look at it. And before we move into the presentation, I just want to remind you that if you have questions, please enter those in the Q&A box, and we will be monitoring that, and we'll try to get your questions answered. And thank you so much for attending.
Here, Mark, I'll take.
I'm sorry, did I need to do something to start the video, Bob?
Yeah, just click it one more time and then don't touch it.
Hi, this is Larry Kuehn. I'm going to be the first of a series of presentations on low-pass sequence data and its use in genetic evaluation. And I'm going to be introducing a joint project between the University of Nebraska at Lincoln and the U.S. Meat Animal Research Center. My topic will be just the introduction of what the project will entail overall. And after me, Warren Snelling will be talking about some preliminary results with low-pass sequencing and its use in predicting genotypes as a potential for replacing the array data that most of us currently use right now. Mark Thallman will follow that and be bouncing off some ideas of what we think we could start to use with this type of data in actual genetic evaluation programs, and where are some of the pathways that we'll be going forward with as we develop this system. So as most of you are aware, currently genetic evaluation relies on genotyping arrays to produce genomically enhanced EPDs. These genotyping arrays generally have between 20,000 and 100,000 different markers, depending on what genotyping array is being used by different breed associations or different genetic service providers. And these arrays are then imputed to a certain set of markers that are utilized as the primary tools for inserting inserting marker information into genetic evaluation. This insertion into forming genomically enhanced EPDs is done using a single-step approach, generally using methods that form a G matrix as a replacement for a pedigree relationship matrix. And most of the time, these approaches are unweighted, such that each of the markers that are being used in the set to make the G matrix have an equal contribution to that G matrix. These markers may or may not be based on a reduced set of markers. There are current evaluation programs that use approximately 50,000 markers, analogous to the original 50K marker data set that's been available for a number of years now. There's other programs that are now using somewhere between 2,000 and 4,000, I believe, markers to make G matrix off of that reduced set, and those markers have been chosen to be more beneficial to the genetic evaluation, higher accuracy than using all markers simultaneously. This approach rarely takes advantage of other knowledge in the markers that might not contribute to the phenotypic information alone. For instance, most of the time this approach does not take advantage of knowledge of variants that are functional or other possible causal variants that could be playing a role in the phenotypes that we're interested in genetic evaluation. So what are functional variants? Functional variants are based off of tools from gene annotation that actually help us understand what kind of polymorphisms are contained within coding regions of genes on the genome. Right now, most of the markers that have been chosen for these genotyping arrays have been chosen such that the markers would be somewhere in an intermediate frequency and approximately equally spaced across the genome. So choosing markers that actually may code for gene function has generally not been a goal. One of the exceptions to that was the F250K chip developed in collaboration with the University of Missouri for a grant project on facility there, where they were actually looking at protein coding regions and polymorphisms that would change the protein or actually cause loss of function in proteins such that the proteins were not actually formed. So basically, the functional variants are a tool that we could use to actually look at what these polymorphisms might actually be changing in the expression of genes and actually see if some of those places where proteins change or where they're not created at all might actually be impacting our trait complexes in genetic evaluation. So as we go into this, we're not going in blindly. We've looked at the F250 chip on some of the data here. This is an analysis from Warren Snelling, and he looked at polymorphisms that we were seeing in the germplasm evaluation, which is GPE in this slide, at the US Meat Animal Research Center. And he did a series of analyses, one of which was based on using all markers on the F250 chip that were shared with the original 50K genotyping array, of which there were around 33,000, almost 34,000. And if he used all of that, he saw a good genomic heritability with that and some transferability of results from GPE to some other populations. The SFA is a selection for functional alleles. It's another population we have at the Meat Animal Research Center, made up of some composites and of purebred Angus. He checked it in red Angus populations that we had some availability with from the Weight Traits Project, and some simmental animals that we also had from the Weight Traits Project. And you can see, yes, results did transfer. We were getting accuracy in all three of those populations when we used everything. But if he did a preliminary analysis and only picked markers that had significant GWAS, genome-wide association study results in the GPE population, the heritability in the GPE population dropped a fair bit, but we actually did as well or better using that same G matrix off of those reduced sets in these evaluation populations. So accuracy was actually increased. And even more interesting with this trait birth weight, when we used 12 of the highest effects, functional variants that were also selected for having low LD with other functional variants that were significant, so actually representing unique signals, we did just as well in terms of accuracy being transferable across the population. Indeed, one marker alone actually explained a huge amount of variance, and that's a known growth marker in CAPG that's on chromosome 6, I believe. So small sets of functional variants can really explain meaningful phenotypic variation within and across populations. And obviously this is an easy example, birth weight. We haven't necessarily reproduced these same types of results on all trait complexes. But what this does is it gives us hope that trying to actually trim down sets of markers into functional sets of markers that may affect gene transcription and translation could actually be a useful tool to selecting which markers may be the most important in our genetic evaluation program. So while the F250 chip did give us a good idea of how functional variants might be very usable in genetic evaluation. There are some inherent problems with the F250 chip alone. So F250 basically implies there's 250,000 different markers on that chip, but some of those were very, very low frequency in our US MARC populations, which is a very diverse population with a lot of different breeds represented. We were only able to see actual variation in allele frequency instead of a fixed allele in about 120,000 of those variants. So that's quite a bit lower. And if we were targeting loss of function genes that were on that original chip, there were only 300 or 703 different loss of function allele changes of the original targeted 50 or 5,751 loss of function polymorphisms that were designed on the chip that we actually saw anymore. And that only represented around 651 genes. So a substantial drop from the original design of the F250. In terms of actual markers that cause protein changes, we dropped from almost 95,000 designed on the chip to around 32,000 that remained and as being variable in our population, and only around 15,000 potential regulatory SNP remain. So a lot of different genes are missing. If we're only representing around 651 and a little over 10,000 genes in the whole genome, realize that there's at least twice as many genes of that generally thought to be on the bovine annotation. So we're missing a whole lot of genes that if they weren't producing the same proteins you would expect from that gene, we may not be seeing what we would expect. So this low pass sequencing that I'm talking about has a new potential to help us out. Basically what this process is doing genotyping instead of by arrays, doing it by sequencing animals at a low coverage. So it's a lot cheaper. And when we sequence across the genome like that, we're able to identify somewhere around 40 to 60 million different variants on the whole genome, and those variants being polymorphisms that perhaps we could exploit down the road. And those are found in potentially in all of those animals using this low pass. The cost has been scaled down. We don't need even 1x coverage per animal. In fact, what Warren will show you later is using coverages that are closer to a 0.5x coverage or less. And basically what's done in order to do this, that wouldn't be enough information by itself, but we're able to use reference haplotypes from fully sequenced animals with higher coverage to identify haplotypes that are out there in all of the different breeds. And based on that haplotype library and reference, we can impute to these 40 to 60 million variant polymorphisms that are spaced across the whole genome. This is also titled low pass or skin sequencing sometime, and we're seeing accuracy of genotype calls upward of 99% on many breeds, and Warren will cover this in a lot more depth in the next presentation. So currently we're entering into a project with the University of Nebraska, Lincoln, and the current objectives of this project then are enhancing the portability of genomic predictors and increasing the accuracy of genomic predictors using low-pass sequencing, and basically accomplishing these goals by evaluating the use of that low-pass sequencing and the large amount of markers that we're seeing, polymorphisms that we're seeing in that, and using those in genetic evaluation systems. So the current plan is to increase genotyping on both populations, on UNL populations, and two different populations that were already mentioned earlier, the germplasm evaluation program, or GPE, and the selection for functional alleles population, or SFA, and basically evaluate accuracy gains from evaluating new marker sets from this low-pass sequencing. And we'll be genotyping all of these populations using a combination of low-pass sequencing, which still isn't quite as cheap as buying genotyping arrays. And so we're basically going to take representative animals and use and run them with low coverage sequencing, and then fill in the rest of the animals with a lower density array and impute low coverage sequencing polymorphisms to the whole population using standard pedigree imputation and haplotype imputation software programs. Right now, we're targeting around 5,000 animals a year in each of the two populations. Part of UNL's is based on an earlier Nebraska beef systems project, and that includes all UNL cow herds as well as animals entering UNL-owned feedlots. We'll be genotyping another 5,000 animals from US Meow Research Center every year, again targeting those two populations and any commercial populations we are observing at the US Meow Research Center that could have some important phenotypes to follow up with. You guys have seen slides like this if you've seen us talk about the germplasm evaluation program. And just to see, as you know, we have a whole lot of traits that we typically measure in this program and report back for getting breed differences or genomic effects on several of these traits. We want to evaluate many of these because many of these are economically relevant traits that are not that well represented in genetic evaluation programs now, and help maybe gain a foothold to make some of these traits that are currently novel in genetic evaluation become less novel by showing that there are places where a little investment in phenotyping might be worthwhile for the breed associations. So analysis of all of this data is not straightforward. We've talked a lot about the number of markers being way over the number of animals when we first started talking about 50K chips more than 10 years ago. Well, now we're talking about tens of millions of different variants that we're seeing, and there's no way we have enough animals genotyped to actually have the number of animals be greater than the number of polymorphisms. So we need to design strategies that give prior weighting to different marker types, like the functional variances I talked about earlier, or regulatory variants or things like that. Basically classify variants and assign different structures of the total genotypic variants to each of these different classes to see if it's worthwhile breaking up the marker sets into things that we think are more likely to affect the traits we're interested in. This includes funding for this plan that we have, includes funding for research support in a joint venture between the institutions, as well as some outside collaborators that are helping out and have interest in this, including the GeneSeq, who's helping us with genotype, and Gencove, who's helping us with a lot of that imputation using reference populations on these low pass sequencing data sets. Mark Thallman will talk about some of the initial ideas for this program and what we're going to do with analyses in more depth after Warren. So there could be some byproducts from this that might come out. There's some potential beyond just improving accuracy of genetic evaluation for us to start to look at extension, look at those novel traits I mentioned earlier, and what can we do to improve and get genome-wide association studies using low-pass sequencing on several of those novel traits. Basically, the extension of novel traits to genetic evaluation will depend on how successful we are with weight traits that we know are already part of the genetic evaluation system, and realize our primary goal here is increasing the utility of genetic evaluation. So the most important strategy that the whole industry can do to help with the novel traits is to try to make them less novel and improve phenotypic schemes in the industry as much as possible. Another byproduct of this as it comes out is, as we talk about replacing the arrays that are currently out there with low pass sequencing, we will start to understand the imputation and storage requirements for low coverage sequence and be able to help inform those types of decisions with several of the genetic service providers and breed associations. So with that, I'll close this portion of the talk and turn things over to Warren Snelling. Thank you.
Thank you. I appreciate the opportunity to visit with you some today about low-pass sequencing and what we think it might do for genetic evaluation. And sure appreciate everyone who's still sticking around for this final session. Hope you get something out of it. You know, what I'd like to do today is cover a little bit about genome sequencing and more on what we're trying to do to evaluate low-pass sequencing before we put a lot into running low-pass on large numbers of animals. It would be great if we could read the sequence of a chromosome from one end to the other. If we could, 2 passes would get complete sequence of both strands of DNA, and we could determine genotypes of all sequence variation that an individual carries. Sequencing machines aren't quite there yet. Right now we need to cut the DNA into random fragments, then sequence ends of those fragments. There are some long-read sequencers that can read several thousand consecutive bases, but most of what we do is with what we call short reads, you know, somewhere, 50 to 300 bases or so. A lot of what we do in low pass is around 150 bases, and we can align those to a reference genome and identify differences between reads from an individual and the reference sequence to call genotypes. EBV were stronger than agreement with pedigree EBV. Agreement for the F250 and 50K were similar. The small F250.
Mark, I didn't get quite far enough back, did I? Not sure what happened there.
I'm not complaining, I just appreciate the help.
Warren, if you're on, how much further back do I need to go, Warren?
Yeah, let's
go further back, I think, Bob.
Okay. Might as well just go there. Go to the next slide.
There.
Go to.
The next one was where it was hung, right?
I think it was hung on that one. So go to the next one. There. Try that. Lots of bulls at low coverage instead of a handful at high coverage. The higher coverage we could have called genotypes on the bulls we did sequence, but at low coverage we thought we could discover more variation and find the interesting variants and figure out some other way to genotype those variants across the population. The GPE bull sequence project got us thinking about custom chips and targeted sequencing as a way to genotype the interesting variants we were finding in sequence. But then we saw some evidence from low-pass sequencing that really got us more interested in this approach. With the low-pass, we can't call genotypes directly from that sequence, but there's some human work that's shown that it can be quite effective, at least in humans, and with a good reference database. And some initial work on cattle samples we got showed a lot of promise. So we started looking more at low pass to get the genotypes we're really interested in. Before generating low coverage sequence to impute a large number of calves, we wanted to evaluate the approach by downsampling some existing sequence to mimic what we can do with low pass. We did this in collaboration with Joe Pickerel and the group at Gencove. The imputation reference panel that they have assembled includes publicly available whole genome sequence from several sources, as well as sequence that we've generated from the germplasm evaluation project. Right now the panel has almost 1,000 animals representing a wide variety of beef and dairy breeds. The breeds are listed in order of representation in the panel. About 40% is split between Angus and Holstein. And then there are some of these categories where the crossbred and composite includes composite breeds developed in the US and Canada, as well as some of the crossbred animals that we sequenced for GPE. And there's some others that the other category is purebreds that are representing a number of breeds that only have a handful of animals in the reference. There is a pretty good set of Brahman cattle in this, as well as a few of the American breeds, Santa Gertrudis, Beefmaster, and Brangus. Nearly 60 million variants were detected in the sequence of these animals in the reference. According to annotation of the bovine assembly, the vast majority are androgenic or intronic and not expected to affect coding sequence. There are 660,000 that look a lot more interesting, that might actually change the protein, or at least regulate how much of a protein is produced. A handful of those are what we call high impact, loss of function that could either silence a gene or cause a major change in that function of that gene. And then another 300,000 or so are non-synonymous and will change the protein amino acid sequence of a protein. Then about the same number are in regulatory regions that might affect how much of a gene is produced. And then there's some variation in non-coding RNA that may also have a regulatory effect. Our evaluation of low pass started with downsampling bulls we had used in GPE. One bull from each of the cycle 7 breeds: Angus, Red Angus, Hereford, Charolais, Gelbey, Limousin, and Simmental, plus a Brahman, Beefmaster, Brangus, and Santa Gertrudis bull were identified. Between less than half an X up to 2X worth of the sequence we had from those bulls were randomly sampled and genotypes imputed from the downsampled sequence. We also used a set of intensely studied steers having extreme high or low intake and gain on the finishing rations. These steers had been sequenced to about 10x and were downsampled to 1x. The imputed bull genotypes were compared to their genotypes from the high-density chip. Correlations between imputed and chip genotypes were generally greater than 0.9 for the Taurus bulls, and increased slightly with higher coverage. Imputation accuracy was a bit less for the Brahman bull, between 0.98 at 0.4x up to 0.99 at 2x. Lowest accuracy was for the composite Beefmaster, Brangus, and Santa Gertrudis bulls. Accuracy for these breeds might be increased by increasing representation in the reference panel.
There's just a pause here at the end of this particular slide. Let's advance it here a bit and see if we can.
I'm not sure what's going on here, but I don't know what happened when I recorded this. But what this slide is supposed to show is that, most of the Taurus, either purebred or crossbred Taurus steers, we got that accuracy up above 0.99, a little bit less for the Brahman influenced steers, but there were a handful of outliers that we had to take another look at. It turns out that all of these with low genotype accuracy were recorded as twins, except for one animal. And that one animal looks more like a twin than it did in the other steers. We also looked at genomic prediction with these steers. The GPE records without the steers were used to train marker effects on birth weight, post-weaning gain, and marbling score. MBV for the steers were predicted by applying those marker effects to the steers' genotypes, both chip genotypes and the genotypes imputed from sequence. And the MBV were compared to the steers' EBV from pedigree and genomic BLUP that did include the steers' records. Three sets of marker effects were solved for each trait. One was the usable functional variants from the GGP F250 chip. A second set was a subset of a few hundred markers from the F250. And the third set was the 50K overlap between the F250 and high density and F250 chips. Agreement between MBV predicted with these marker effects and the steers GBLUP EBV were stronger than agreement with pedigree EBV. Agreement for the F250 and 50K were similar. The small F250 subsets explain less than the large marker sets, but still explain the substantial amount of variation in these traits. Agreement using the chip genotypes was a bit higher than agreement with imputed genotypes, which is probably because genotypes for some of the chip SNP were missing from imputed genotypes. And one other data set we looked at is something we just received last week and haven't had a whole lot of time to explore. But These were from a set of UNL cattle imputed from half-X sequence. All were purebred Angus or red or black Sim Angus females. At this point, we don't have other genotypes for comparison, but did apply the call confidence metric to the imputed genotype probabilities. Call confidence for these girls is generally lower than what we saw for the steers, but none of these cattle have the extremely low call confidence that might indicate DNA problems like we did find with the steers. So far, everything we've seen from downsampling is pretty encouraging. Imputation from low coverage sequence appears to be pretty accurate. It's not perfect, but one feature of the imputation is that it can recognize where it's not going to impute well, and as reflected in the genotype probabilities. We might improve accuracy by expanding the reference panel, and another round of imputation that might include pedigree to impute the low probability genotypes. With low pass, we can impute genotypes for a comprehensive set of variants detected in the reference panel. Not that we're ever going to use all 600 million in an evaluation, but we can extract interesting subsets to use for the evaluations. Most immediately, the 50K can be pulled out and used with existing chip data, just like we do in current evaluations. As we learn more about which variants are more likely to affect performance, the 50K SNP might be replaced with probable causal variants, reducing the dependence on within-breed LD to enable more accurate prediction across different breeds, crosses, and generations. Right now, the cost of low-pass sequencing and imputation is competitive with SNP chips, but low-pass can provide much more information. Developments in library prep and sequencing costs may reduce costs even more, which could encourage more complete genotyping to reduce bias and increase accuracy of genetic evaluations. Lower cost and more accurate predictions across breeds and crossbred cattle might also help to justify genotyping commercial calves. Commercial data could be included in national cattle evaluation, and genomic predictions on commercial cattle might support calf management and marketing decisions. The big advantage we see for low pass with imputation instead of chips is that we can genotype interesting variants without all the issues related to custom chips. We can implement low pass in GPE and other research populations right away at a fraction of the cost it would take to do a chip with just some of the variants that we'd like to genotype. Even with collaborations, we wouldn't have the volume needed for a low-cost chip. And some of the genotype calls from a chip might be questionable until we have enough data to really train the calls on the chip. With low pass, we don't have to worry about using up chips with limited shelf life or variants that are missing from the chip because probes for those variants are not designable. All this does not mean that we're completely satisfied with imputation from low pass. One particular concern is accuracy of imputing genotypes for genetic defects. We need to know the sequence variants causing the defect, and the reference panel needs to include defect carriers to have any chance of low-pass sequence matching haplotypes with the defect alleles. Lower accuracy of Brahmin-influenced composite bulls and steers suggests a need for more indicants influence in the reference panel. Additional expansion of the reference panel might be suggested by genomic relationships among commercial cattle and the reference panel. Right now we have a summer student working on that using genotypes provided by Neogen and the Gencove reference panel. Eventually, we need a systematic approach to identify holes in the reference. Monitoring genotype probabilities of calves going through the imputation pipeline might indicate groups needing additional reference. We also need to revisit imputation from chip genotypes to sequence variants so we can leverage the existing chip genotypes. For this, pedigree imputation to a large reference panel genotype with low pass might be more accurate than imputing to a relatively small set of sequenced bulls. Overall, we're encouraged by the possibilities low pass offers for genetic evaluation, but there's still room for improvement. None of this would have been possible without a lot of help. Genomics deserves a big thank you for introducing us to low pass. Gencove has been really instrumental both in constructing the reference panel and developing an imputation pipeline. You know, we've received some challenges and ideas and support from GeneSeek, and then the whole entire operation at Clay Center, everyone from the cattle crews through the core lab doing the sequencing, have made all of this possible. Thank you.
Hi, I'm Mark Thallman. I want to visit with you about opportunities for low-pass sequencing of pedigreed populations and how it may fit into genomic evaluation. With current genomic EPDs, we assume markers are spread evenly across the genome in intermediate frequencies or are selected from such markers. We assume some markers may directly affect traits, but know that most do not. We assume causative variation is closely associated with markers. We assume all genotype animals either have or can be imputed to a common set of markers. And one thing we can be pretty confident of is that current genomic predictions are more accurate than predictions without genomics. With our current approach to genomic EPDs, we could expect to get a limited increase in accuracy from improving utilization of the markers that are on the current chips. We could expect to get an additional limited increase in accuracy from increasing the number of markers of the same type as what are on the current chips. But the high-hanging fruit is causative variation not on current chips that often has low minor allele frequency. There are millions of candidates and only limited opportunities for prioritizing them without having genotypes to evaluate their effects. Nonetheless, Warren has shown benefits of screening putative functional variants from a relatively small subset of the entire pool of such variants. Our current default approach to improving the accuracy of genomic predictions is something like the following. We sequence influential bulls within the population. We look for variants within that sequence. We impute that sequence to descendants of these influential bulls using chips of a variety of densities. Within that data, we identify the most promising sequence variants to improve accuracy of the predictions. We use functional information and preliminary associations with traits where possible. And then we develop new chips that include these promising new variants. We determine which of the promising variants appear most predictive in our evaluations. We include those most predictive variants in the next generation of prediction models and in future chips, and then we repeat the process. If this looks hard, that's because the high-hanging fruit is most of what is left to do, and it is hard. But Matt Spangler calls this iterative redesign of chips untenable when considered in the context of low-pass sequencing as an alternative. Low-pass sequencing is sequencing a random sample of the genome of an animal to low coverage in lieu of genotyping a specific set of markers. The short-term goal is to impute to the standard set of markers used in the current analyses at cost that's competitive with genotyping. The intermediate goal is to identify markers that are more predictive of important traits And the long-term goal is to replace genotyping by imputing an entire population to full genomic sequence. By comparison, chip genotyping provides high accuracy and call rate without imputation, whereas low-pass requires imputation for both. Chip genotypes give us both the paternal and maternal alleles, whereas low-pass may impute the paternal allele but not the maternal, or vice versa. Chips are focused on genotypes, whereas low-pass is focused on haplotypes, and chip genotyping is a mature technology, whereas low-pass is in early stages of development. There are legitimate concerns over low-pass sequencing. First is how it will integrate with existing SNP chips and the subsets of them used in current genetic evaluations. Warren has shown that it is feasible to get good marker genotypes, but that there are limits on that. Secondly, will genetic defects and other must-have variants such as POLD and red-black be reported reliably? But there are several approaches available to enhance the representation of specific loci within the library. Low-pass does require imputation to produce a useful result, but imputation is already part of the genomic evaluation pipeline. The imputation required for low-pass is more sophisticated than that currently used, but Warren has shown that also is feasible. And finally, will it work for parentage determination? Well, SNP chips are great for parentage determination, but low-pass is going to be even better. So why consider low-pass sequencing? It will make the process of SNP discovery, promising variant identification, adding them to the evaluation, validating them in field data, getting rid of the dropouts, returning to SNP discovery, and repeating far more seamless, continuous, and less time-consuming than iteratively redesigning SNP chips. The current cost is somewhat greater than that of 50K chips, but it may decrease to below SNP chips. SNP discovery will be far more thorough than if it's limited to higher coverage of relatively few influential bulls. I'm going to take you through an example that I hope will help you better understand the differences between chip genotypes and low-pass sequence, and also understand how we can generate near-complete sequence from low-pass data. To do that, I'm going to use a hypothetical pedigree comprised of a founder, sire, and dam, 2 full sib progeny of them, and a son of the 2 full sibs. I do not mean to imply that the actual animals of which these photos were taken are related. To the right of each photo is a hypothetical sequence of that animal with the sequence inherited from its sire above this sequence inherited from its dam. We're going to take a microscopic view of the genome and focus on a hypothetical region spanned by 3 consecutive markers of a 50K chip. But everything we do within that small region would be happening simultaneously throughout the remainder of the genome and in regions spanned by another 50,000 or so markers scattered throughout it. To help remind us of that, there are ellipses at the ends of the sequence that represent the sequences between the ends of the region and the ends of the chromosome that is on. The total length of the sequences represented by the 2 ellipses would be on the order of 100 million bases for an average bovine chromosome. By bases, I mean the fundamental building blocks of DNA, which take 4 forms that we represent by the letters A, C, G, and T. This region of the genome that we're going to focus on would typically be about 100,000 bases in length, or 0.1% of the length of the chromosome on which it resides. The locations of the 3 50K markers are highlighted in yellow, and we also assume a variant that directly affects a trait of importance to us that is highlighted in blue. Now we're going to zoom in on a few hundred bases that surround each of the 50K markers in the causative variant. There are 4 such segments on the slide, separated by tildes. Each tilde represents constant and variable sequence that is important but too long to represent on the slide, generally greater than 10,000 bases in length. Within each of these several hundred base segments, we will further focus on the positions that are variable within our population. They are the ones that are represented by letters. And then we have the dashes, which represent sequences the same within column but different between columns. The differences between columns are important in that they help us determine where in the genome this sequence resides. Before we leave this slide comparing chip genotypes with low-pass sequence, I want to ensure that everyone understands some of our jargon. The genotype is the pair of bases that an animal has at a specific position in the genome. A haplotype is a string of bases along a chromosome that all have the same parent of origin. So the genotype of the first bull at the first marker is GA, His paternal haplotype is the row of blue letters, and his maternal haplotype is the row of green letters. The 50K chip gives us the information in the yellow columns, whereas low pass will give us most of the information on the slide. I'm going to go through the example fairly quickly and try to emphasize the main points that I want you to take home from it. There may be a few of you who want to watch it again at your own pace and get into the weeds and gain a deeper understanding. If you fall into that category, please read the disclaimers on this slide. This slide shows a representation of low-pass sequencing reads for the first 3 animals in our example. Each contiguous string of letters and dashes represents one sequencing read, which would typically be several hundred bases in length. It's not possible in this diagram to show how spread out these reads would be across the genome, so see Warren's diagram for a better idea of what that would look like. Warren mentioned that having a good panel of reference haplotypes is important for imputation of low-pass data. Here we have an example set of reference haplotypes that are comprised of 3 haplotypes present in our founder individuals and 1 haplotype in red from outside the pedigree. But the reference set is missing the cow's maternal haplotype, which I've shown below the reference panel. In the middle of the slide, we have the low-pass reads from the founder cow from the previous slide. And we can see that the reads that are purple match exactly with the purple reference haplotype. So we just copy the purple haplotype down from the reference panel to the cow's imputed genome sequence. And we see that the 2 gold reads on the left half of the slide match exactly with the red reference haplotype. So we copy that down to the cow's imputed sequence below, but we find that the gold reads on the right half of the slide don't match at all with the red haplotype or any other haplotype in the reference panel for that matter. So we are not able to impute the sequence on the right half of her maternal genome, so we can fill in the positions that were directly read by the sequencer, but we are unable to impute the other positions, so we use dots to represent the unknown bases in those positions. Careful comparison of the red and gold haplotypes reveals 2 positions in which they differ, and that introduces 2 imputation errors in the cow's maternal haplotype. We will track that as we go through the example and see later how to resolve it. So what if we use the pedigree in imputation? If the parents have reasonably complete and accurate sequence imputed, we can do a lot. To demonstrate this, we will add sparse coverage to the bottom 2 animals. By sparse, I mean lower coverage at lower cost than low pass, with reads correspondingly spaced farther apart. To start the process, we need sequence reads that can be assigned unambiguously to only one of the 4 copies of this chromosome segment within each animal's 2 parents. A fraction of reads meet that criteria, but those that do tell us whether this region of the genome was inherited from the animal's sire or dam and whether it was the parent's paternal or maternal copy. In doing so, it tells us which reference haplotype the read should be assigned to, and by extension, which other animals in the pedigree are identical by descent to it. The reads shown for the bottom 2 animals meet this criteria, with one exception that I will describe in a minute. Fortunately, we only need relatively few reads this informative to do powerful pedigree-based imputation. The power of this imputation is hidden in the colors of the ellipses at each end of this region. They indicate identity by descent in the region immediately adjacent to the one being considered. In the case of the black cow, suppose it reads that can be attributed to her sire in both regions adjacent to this one in the diagram consistently match his green maternal haplotype and not his blue one. Then the ellipses on each end are colored green, and it is safe to assume that everything between them is from the green haplotype because the odds of a double recombination would be on the order of 1 in a million. If the ellipses are different colors, a recombination occurred and things are a bit more complicated. But that is only expected in about 1 out of 1,000 regions of this size. This approach does depend on the sequence haplotypes of the parents being sufficiently complete. Here the progeny haplotypes are imputed by copying the appropriate parental haplotypes and highlighting them in light green. The paternal haplotype of the red bull has a new crossover in addition to the crossover that occurred in his sire. The purple and green reads represent the closest points to the crossover where parental origin can be determined, so the interval between them is colored gray to indicate ambiguity of origin, and only the positions where the 2 parental haplotypes agree are filled with base calls. The sequence data generated by sparse coverage provides information to fill gaps and fix errors in the ancestors. Essentially, wherever haplotypes are identical by descent, information can be shared between them. In the case of the imputation errors, information from two sources was contradictory, so both were set to missing to be resolved later with yet more information. After having sequenced 3 animals with the gold haplotype at low or sparse coverage, it could be added to the reference panel. Even with some gaps and potentially undetected errors, imputing animals identical by descent to these with the gold haplotype in the reference would probably produce more accurate results than imputing with the red haplotype as the best option for them. This example was contrived to generate problems and to show how they could be solved. Both were exaggerated due to the constraint of having room to show 5 animals with 24 variable positions across a region spanning 100,000 bases. What this example completely failed to demonstrate, and I have to ask you to imagine, is the power that a couple dozen progeny per bull with sparse coverage would have to fill gaps, resolve errors, and identify new haplotypes to be added to the reference. So off-the-shelf low-pass works amazingly well. Could work better and be less expensive with pedigree information. The advantages are far greater if the entire herd or population is sequenced than if just a few. And low-pass captures far more genetic variation than current chips can. Most sequencing of cattle has made the simplifying assumption of a common reference sequence, which has usually been of one particular Hereford cow. Although we've known for a long time that there's variation in the structure of cattle genomes, until recently it's been one of many complications that were not feasible to deal with. My colleague Tim Smith is leading a collaborative effort to characterize structural variation by long-read sequencing the genomes of individuals representing many diverse breeds of cattle, with the objective of constructing a pangenome of cattle. This slide attempts to illustrate the concept with hypothetical structural variation between and within several diverse breeds. The pangenome is a collection of all sequences that can be found in cattle, while the core genome is a set of sequences that all cattle share. Markers that are candidates for chip genotyping are typically screened for call rate, which is the proportion of animals for which genotypes that follow Mendelian segregation can be reliably produced. This would exclude most markers outside the core genome, so most markers on current chips are probably in the core genome. In contrast, all the DNA in the genome is subject to low-pass sequencing. When it comes to structural variation, we're just getting started in cattle. There's much more we don't know than what we do know. We do know some genes that vary in copy number. It seems likely there are at least some genes that are expressed in some animals and absent in others. Such genes seem likely to contribute to functional variation. The problem of missing heritability is that there's some variation that we know is heritable, but we can't find it with current genomic approaches. It seems likely that structural variation contributes to the missing heritability. Structural variation is detected much more effectively through long-read technology than with the short reads used in low-pass. But once detected and added to reference haplotypes, it should be feasible to impute structural variation with short-read low-pass sequence generated currently. And so that emphasizes one of the advantages of low-pass, which is that we don't need to know what we're looking for before we run the assay. And that's a huge advantage. Larry told you that we're initiating a pilot project in collaboration with the University of Nebraska, in which we're going to add low-pass sequence to populations at UNL and several populations at US MARC. And I'm going to give you a little bit more detail on what we're doing within the germplasm evaluation project. We have already sequenced almost 400 sires that are influential in the project, comprising 20 breeds at 2 to 4x depth of coverage. Those have already contributed to the reference haplotypes that are being used along with other sources. And much of that sequence is on sire-son pairs to enhance the efficiency of haplotyping. We've genotyped much of the GP population already with chips of various densities. We've prioritized 3,000 animals for low-pass sequencing and several thousand others for additional low-density chips. The animals designated for low-pass are those expected to fill the most holes in the reference haplotypes. When we get all this data back, we'll evaluate the quality of imputation. We'll do additional sequencing to fill the most important holes in the reference. And we'll develop analyses to utilize the imputed sequence to identify predictive markers not on the chips and improve genomic predictions. So one of our objectives is to provide information that could inform decisions about whether, when, and how the beef industry might transition into low-pass sequencing for genetic evaluations. And so our best guess at a strategy for that is, obviously, we would begin with a collection of reference haplotypes that's appropriate for the populations to be evaluated. Then use low-pass instead of chips as it becomes cost competitive to do so, or can be demonstrated to provide sufficient accuracy to justify the cost. Verify that the concerns listed above are addressed, especially that we're continuing to get good genotypes for the marker loci that would still be involved in or required for the genetic evaluation. We want to evaluate quality of imputation and accuracy of prediction, and then collect additional sequence on those individuals that would most effectively fill the most important holes in the reference sequence. Our short-term goals under this strategy would be to keep the current marker sets and models until low-pass comprises a substantial proportion of the data, and to monitor the quality of imputed genotypes for those markers. Then we take a two-pronged approach, the first of which is to identify and sequence influential ancestors, which, if low-pass sequenced, would provide imputed sequence to the greatest number of phenotyped individuals. And the second prong is to use non-production genetic evaluation runs to continuously screen for variants not in the model that have the greatest predictive ability, and continuously but gradually add loci with the greatest predictive ability to the model and drop those that are least predictive. That would include loci outside the core genome, and we would give extra emphasis to functional and putative regulatory SNPs. Simultaneously with that, we would impute the genotypes of loci that are in the production genomic evaluation model but not included on chips back to those animals that are genotyped only with chips, so that they could also be evaluated to some extent based on those additional loci. Okay, we're just about to the conclusion, so if you've hung in with me this far, don't quit if you happen to hear some jargon in this slide that's new to you. But I think we're going to have to, in the long term, replace the genomic evaluation model that we're currently using. And one possibility would be a hierarchical model in which part of the model relates a haplotype layer to an unobserved gene activity layer, which would be informed by prior probabilities of variants influencing gene product function or gene expression level. And the default assumption would be that variants not in the immediate region of a gene affect the gene only through their own gene products. The second part of the model relates gene activity layer to the phenotypic layer of many traits with priors based on physiological gene networks and other concepts from systems biology. The gene activity layer would not be trait specific and would be informed by low-pass RNA sequencing of many tissues under various conditions, and also by proteomics, metabolomics, low-pass metagenomics, and other physiological indicator traits. The low-pass RNA sequence data could then replace some of the coverage requirement for low-pass genomic sequence. And under this approach, dominance and epistasis would be expressed at the gene activity layer. This approach reduces the dimensions of the parameter space and incorporates many additional sources of information relative to the current model in which each variant is potentially and separately related to each trait. I'm sure there are many other possibilities for ways to improve the genomic evaluation model. Many of them probably have not yet been thought of. One of the reasons I think we're going to have to reevaluate or approach the genomic evaluation is the P much greater than N problem that Larry alluded to in his part. What that is essentially is that the number of parameters that we need to estimate, which is denoted by P, is much greater than the number of observations denoted by N that are available to help us estimate those parameters. So in the context of genomic evaluation, what that means is that we have many times more marker effects than we have animals. This problem is sometimes also called overfitting or model overfitting. And if it's not accounted for, it causes predictions to appear much more accurate than they really are. There are many ways to deal with it. They're pretty technical, so we won't cover them here. This was a serious problem in the early days of genomic EPDs based on SNP chips, especially when we had 50,000 marker effects to estimate, and many breeds had only 1,000 or a few thousand animals genotyped with those 50,000 markers. It's become much less of a concern recently, as several breeds now have substantially more animals genotyped than there are SNP available for inclusion in the model. And there are also arguments that the genomic evaluation models currently being implemented are not susceptible to this problem. But as we consider selecting markers from tens of millions of candidates, P much greater than N is going to reemerge as a problem that we have to deal with. Nonetheless, our best chance to improve accuracy is to consider all variants, so we will have to return to dealing with the P much greater than N problem. Finally, in 2015, I presented a poster arguing that successful widespread utilization of what's now referred to as low-pass sequencing was dependent on technological advances in two areas. First is cost-effective methods for construction of sequencing libraries, and second is the algorithms, data structures, and software to efficiently impute low-pass data to genomic sequence throughout populations. I remain as convinced as I was then that those are the two impediments to implementation, and I'm really encouraged that although much work remains to be done, Warren demonstrated substantial progress on both fronts, and that low-pass is competitive. I'm really optimistic that there are immense opportunities to make further progress on both of those fronts. So in conclusion, there's far more information in an incomplete and imperfect view of the majority of the genome, which is represented by low-pass sequencing, than there is in a near-perfect view of a minute fraction of the genome as we can get through chips.
Okay, do we have any questions? Well, since we don't seem to have any, I'm going to move on to Daniela's presentation.
There's one question, Mark.
Oh, is there? Okay, good. Thank you. Is it expected that we will be able to impute the large populations of commercial animals genotyped on the commercial chips up to the sequence level in a routine manner?
I've just been talking with Gencove about this, and what they say is, I think it's safe to say that Gencove is pretty comfortable with scaling processing of data to the current scale of data throughput. The phrase we like to use is embarrassingly parallel, but also the analysis itself is pretty efficient. Time of imputation is similar to chip genotype calling and much less than the wet lab production steps. So that's thanks to Jesse Hoff at Gencove.
Okay, thank you, Warren. So I think you understood the question better than I did, so thank you. Any other questions before we move on to Daniela's presentation? Oh, okay. Sorry, I was having trouble advancing the slide. So our next presentation is on a fairly similar topic, increasing accuracy of genomic predictions from SNP chips to sequence data. And it is presented by Dr. Daniela Lourenco of the University of Georgia. And she is an assistant professor in animal breeding and genetics at the University of Georgia. She was born and raised in Brazil, where she earned her MS and PhD degrees in animal breeding and genetics from Lourenco State University. Daniela has been working in this field since 2004, has published over 300 scientific papers and proceedings, including over 90 refereed journal publications. Her research is focused on improving livestock production using genomic information, developing methods for genetic evaluation, and addressing issues related to the implementation of genomic selection in beef and dairy cattle, poultry, swine, and fish. She's been involved in the implementation of single-step genomic evaluation for some of the American and Canadian beef cattle associations, and has also been actively working with several U.S. and international breeding companies. So,
let me see if I can, okay, I can do it.
You have it, Daniela?
Yeah, I do have it.
Very good, thank you.
Yeah, thank you. So thanks, Mark, for giving me the opportunity to talk today. And I would like to thank everybody that is still connected to this conference. So I know it's pretty late in some parts of the country and in other countries. But anyways, I'm very happy to be here today and talk a little bit about my experience on using regular SNP chips and also sequence data for genomic prediction. And I will try to keep this talk really simple. And there are three reasons for that. One reason is today's Friday. The second reason is that I'm the last speaker in the last day of the conference. And the third reason is that we don't have a barbecue reception, so you can feel the pressure that is on me, right? So, okay. We started using genomic tests back in 2009. So last year we celebrated 10 years of the implementation of genomic evaluation, which is just to use DNA information to help us to identify the best animals. So if you think that this idea is quite new, I need to tell you that it's not new. And I always like to show this. So this paper here shows that in 1983, two guys said that we could use DNA information to construct genetic relationships, to do parentage determination, and to identify genes or QTLs, or quantitative trait loci. So they told us that DNA information could help us in animal breeding and also plant breeding. But then what kind of genomic information are we using? We all know that we use SNPs. So SNPs are just changes in the letters in the DNA. And since the SNPs were first reported, and this was in the first draft of the Human Genome Project, the SNPs, they became the bread and butter of DNA sequence variation. So they're highly used in animals, in plants, and also in humans. So if we think about animals, so if you think about like livestock animals, if you think about poultry, if we think about aquaculture species, so how many SNPs are we looking at? So we are looking at about like, we are looking at about like from 3 million to 3,000, sorry, to 800,000 SNPs. And how do we do that? So we need to collect a sample. So we collect a blood sample or hair sample or tissue sample. We send this sample to the breed association. The breed association is going to send this sample to the lab. So in the lab, the technicians are going to extract DNA, and they run the DNA into this SNP chip here. And in this SNP chip that I'm showing here, we can run 24 samples at the same time. And then after we run the DNA into those SNP chips need to be scanned in some machines here. So we have a scanner that's going to read the genotypes for each SNP. And what is interesting here is that if we have heard about 23andMe or Ancestry, so they do genetic tests for humans, and the idea behind this genetic test is exactly the same idea behind the genomic test that we do for animals. The only difference here is that the SNPs are species specific. So for animals, we have certain SNPs, for humans, we have different SNPs. So the SNP chip is different, but the idea behind it is quite similar. So once we have the results from the lab, so we have the genotypes for the animals, what do we do with those genotypes? So the results from the genomic test. We all know that we just include this information, so we include genomic information in our evaluation system. So we have pedigree performance, and also we have progeny performance. So we include this genomic information, and then we have what we call genomic enhanced EPDs or genomic EPDs. And we all know that genomics is an extra piece of information that helps us to increase accuracy of EPDs, right? And because this increasing accuracy is so considerable, genomic tests, they were adopted by pretty much all livestock species and aquaculture and poultry species. So, and because of that, the number of tests that we have right now can be quite considerable. So if we look here at like farm-raised fish population, so they may have about from 2,000 to 10,000 genotyped fish. If we look at chicken populations or pig populations, we may see some companies that have about 50,000 animals genotyped per line. If we think about sheep populations, we may see populations, especially if we have like multi-breed populations, we can have like over 100,000 genotyped animals. And here in the US, for jerseys, they have over 460,000 genotyped animals. And if we think about beef cattle, the largest population genotyped is Angus. So in this case, they have almost 800,000 genotyped animals in the database. But still, the population with the largest number of genotypes is Holstein. So they have about 3.4 million genotyped animals. So we can see a quite considerable number of genomic tests for livestock animals, for poultry, and for aquaculture. But then what do we gain by adding this genomic test? So here, just to remind you that those numbers are mainly based on regular SNP chips, right? So here in this example, I have the gain in accuracy by using genomic information, and this is for Angus. So we have here the gain for birth weight, weaning weight, and post-weaning gain. So in blue, we have the accuracy when we did not use genomic information. So this is just based on traditional evaluations before genomics. And then in green, we have here genomic evaluation. So this is for single step GBLUP in 2014, when we had about 52,000 genotyped animals. And then in golden, we can see here the accuracy when we used about 335,000 genotyped animals, and this was back in 2017. So if we compare the accuracy like from the traditional evaluations and when we use about 52,000 genotyped animals, we saw an increasing accuracy of 25%. But then when we added more genotyped animals, like we had 335,000 genotyped animals in the system, we saw an increasing accuracy of 36%. So we saw a considerable increase in accuracy. And why is that? So we are going to see increasing accuracy when the genomic information is non-redundant in the system. So if this information is not giving exactly the same information as the other source of information that are in the system. So just to illustrate what I'm talking about. So let's say that we have a bull with a lot of progeny records. So, and the accuracy of this bull is 0.99. So if we genotype this bull, we are not going to gain in accuracy. We're not going to see an increase in accuracy because the EPDs are accurate enough. But then if we genotype a bull that has no progeny with records at all, in this case, we are going to see a considerable increase in accuracy. And this is because genomics is non-redundant with the other pieces of information that are in the system. So in this case, genomics is acting exactly like as we are adding progeny records. And I want to show you here the equivalence between doing a genomic test and the number of progeny. So if we genotype a bull, and then the gain that we can have in accuracy is the same as if we add 27 progeny with records for weaning weight. But then for other traits like mature height, the gain in accuracy may be equivalent to adding 9 progeny with records. So, and this progeny equivalent is actually dependent on heritability of the trait. So if the trait has high heritability, the equivalent is lower. If the trait has low heritability, the progeny equivalent will be higher. So I just want to show you here a kind of extreme example. So in Holsteins, if we genotype a bull, and then we evaluate a trait that is daughter pregnancy rate, and this trait has very low heritability. Heritability is 0.04. So this genotyping an animal for this trait is equivalent to adding 131 daughters with records in this case. So genotyping is the same as adding progeny records for 131 progeny. So we can see that this is pretty considerable. So we know that using the regular genomic test, we are doing a very good job already. And if you ask my opinion about that, I think genomics is actually the best thing that was invented since sliced bread. So no question about that, right? But then the question here is, so, okay, we see that we can increase accuracy. And then what do we gain by increasing accuracy? So to answer this question, we can just look at the Breeder's equation here. So R here is the accuracy, and L is the generation interval. So another thing that genomics can do for us is to help us to better identify the best animals earlier in life. So in this case, we are reducing generation interval. So when we increase accuracy, which is R, and we reduce generation interval, we are going to see an increase in accuracy here. So this is what we gain by adding genomic information, right? And then because we started using genomic tests like some time ago, we do have some examples to show what happened with genetic gain since we started using genomic information. And earlier today, we saw John Genome, he showed some trends for beef cattle after genomic testing started being used. And then today, I brought here two examples that are not in beef cattle. So the first example is in dairy cattle. So some researchers from the USDA and also from some research institute in Mexico, they showed that, so what they did, they evaluated for US Holsteins. So the genomic evaluation started being used for US Holsteins in about like 2009. So in 2015, they evaluated the gain after genomics was implemented. So what they saw here was a gain. So the they saw that the genetic gain increased from like 50% to 100% for yield traits. And for traits with low heritability like fertility, this gain was like from 3 to 4-fold. So we could see a considerable increase in genetic gain for dairy populations. And another example that I want to show here is from the Pig Improvement Company, so from PIC. I asked my colleagues from PIC to draw this graph for me. So here we have the graph for genetic trend. In the y-axis, we have some selection index that they use to select the animals. In the y-axis, we have birth year and month. So it starts here from January 2006, and it goes up to May 2019. So here, this red line here shows the trend. So we can see that genetics is improving, so it's going up. And then the blue vertical line here shows when genomics started being used. So here, when they started using single-step genomic blood for genomic evaluations, and this was at the end of 2013. So if genomics had no impact at all, we would see this red line just following this gray line here. But what we actually saw was that this red line shifted up. So this tells us that we have an increase in genetic gain. So this tells us that genomics is actually increasing the genetic gain. So if genomics had no impact, the average for selection index would be about like 70. But then since genomic started being used and it has an impact, we saw here a 2-fold increase in genetic gain. So this is the benefit that we can see by using genomic information. So we can see here that we had an increase in genetic gain, but then some people may look at this graph and just say, oh, okay, so some of this gain may not be due to genomics. And actually I was asked about that already. So I went back to my colleagues and I asked them, so can you try to identify the percentage of this change that was due to genomics? And then they told me, okay, about 95% of this change was due to genomics. So this is because there was an increase in accuracy and a decrease in generation interval. And if we are talking about increasing accuracy, what do we need to do? So we need to, this accuracy is going to depend on the number of genotyped animals. So we need to have a lot of genotyped animals. It depends on the number of performance records that we have in the system. It depends on the heritability of the trait. It depends also on how informative the genomic test is. And we've been using a genomic test that has about 50,000 SNPs in beef cattle, but it can range, so it has a range. It can be like less than that, a little bit more than that, but on average, let's say about 50,000 SNPs. But then the question is, so is this the genomic test informative enough? Well, we actually don't know. We have an idea. Okay, and I'm going to talk about that later. Okay, another question that we have is, do we have enough SNPs in the test? And also, extra question, are we looking at the right SNPs? Well, just to talk a little bit more about this last question here, if we are looking at the right SNP. So just to illustrate this idea. So the early developed SNP chips, they actually had about 70% of the SNPs that could be informative over like across breeds. So the first 50,000 SNP chip that was developed, so it had 54,000 SNPs. And then if we use this SNP, so this SNP was developed based on beef and dairy breed. So if we use this SNP chip in Angus, we would see only 38,000 SNPs that were informative enough. So the remainder of the SNPs we had to throw away from the evaluation. And this is true also for pigs and for chickens. So in chickens also, like from the 54,000 SNP chip, we could use about 39,000 SNPs. So because of that, and also because of other key points, we decided to try to use more SNPs, right? So instead of using 50,000 SNPs, what about looking at all SNPs in the DNA, try to find all SNPs. So to do that, we need to sequence the animals. And when we sequence the animals, we may be able to find like from 30 million to 60 million or maybe 100 million SNPs, right? And just to remember that in the DNA, in the bovine DNA, there are about 3 billion base pairs. So from 3 billion, at least 30 million are SNPs. And to understand why or how this is, sequence information can help us, or if this is going to help us to increase accuracy, we need to remember a key point here. So the point that we need to remember is that the SNPs, they're just trying to capture information about genes. So, and what kind of information? One source of information, is relationships. So SNPs are just trying to capture relationships at the gene level. So just to understand it better, I got a picture here from Jared Decker from University of Missouri. So in this picture, we have a sire, and it's maternal and paternal grandsire. So it's expected that this animal shares 25% of its DNA with the paternal and with the maternal grandsire. And this information is the information that we use in the regular evaluation, when we do not use genomic information. But then let's go to the next level where we have genomic tests. So we have information from SNP chips, right? Okay. So in this case, we genotype the animal and its grandsires, and we see that this animal shares 15% of its DNA with the maternal grandsire and 25% of the DNA with the paternal grandsire. So in this case, we have what we call the observed relationship. So we actually have the proportion of the 50,000 SNP chips that are shared between the animal and its maternal grandsire and the animal and the paternal grandsire. Okay, so let's go now to the third level. So the third level would be to use sequence data. And what is the idea behind it? What's the hypothesis here? Is that with sequence data, we may find SNPs that give more information about genes. And these may be true if those SNPs, they are close enough to genes, or if they are really giving us information about the genes. And just to illustrate what I'm talking about, so let's say that we have a gene here represented by this green point here. Okay, so let's say we use 50,000 SNPs, and then this is what we may have. So we may have SNPs that are close enough to the gene, so those SNPs are going to give us information about the gene, right? So we may have good accuracy here. But let's say that we just sequence. We have sequence information. We have information from about 30 million SNPs. So in this case, we can see that the SNPs, they are covering larger areas of the DNA. And we have SNPs here that are even closer to the gene. So if those SNPs that are closer to the genes, they're giving us extra information, so information that is non-redundant with the information that is already given by the 50,000 SNPs, we may be able to see an increase in accuracy. However, if the orange SNPs and the blue SNPs, they're giving exactly the same information, a gain in accuracy is unlikely. So in this case, we pretty much are not going to see an increase in accuracy. So it depends on how non-redundant this information is going to be in our evaluation system. And we know that sequencing is becoming cheap. But still, what we see is that, and also we heard from the previous talk, that we just sequence a small proportion of the animals right now. And then what we do, we impute this sequence information to the all the genotyped animals. So we have genotyped animals with regular SNP chips. We sequence just a portion of them, and then we impute to the other animals that are genotyped. So it's like just filling the gaps, right? And then there is a paper here, a study published by some colleagues from PIC and also from the Roslin Institute, where so they had some pig populations, so several lines. And then in those lines, they had genotypes like regular SNP chip for about 18,000 to 107,000 pigs. So what they did, they sequenced about 2% of the animals in each population, and then they imputed to 20 million to 30 million SNPs, and they imputed to all those almost 100,000 or a little bit over 100,000 animals. And the imputation accuracy that they obtained here was like from 94% to 98%. So this is quite good, and it tells us that we can have a massive amount of sequence information in a cheap way and also in an accurate way. And then last year, at the end of last year, we joined Roslin Institute and also PIC in this project. So the objective for us is to try to use single-step GBLUB in this case, and then try to see if we can have a gain in accuracy when we include those sequence SNPs in the evaluation. And this is mainly for each line. And then another objective is to try to see how, or if we can have like more accurate multi-breed evaluations when we use this sequence information. And another thing that we are going to investigate also is the persistency of accuracy over generations. So if having information from sequence data will help us to get more persistent genomic evaluations. And then we are going to do that for several lines. Actually, we started doing that, but I don't have results to show here today. So we got this data like with about 110,000 pigs with sequence data, and it's a massive amount of data. And they were imputed to about 20 million to 30 million SNPs, right? But then the question is, do we use all those 30 million SNPs? Well, not really. We don't use those 30 million SNPs because 30 million is too many SNPs to use. And also because most of the SNPs, they're redundant, they're not giving extra information. And also if we use those SNPs, those 30 million SNPs in the evaluation system, we will have some errors that when those errors accumulate, because we have a lot of SNPs, we are going to see in fact a decreasing accuracy instead of an increasing accuracy. So what we do instead, we need to try to mine SNPs that may be more significant. So, and what do I mean by mining SNPs? So this is just like using some statistical methods to try to see which SNPs are related to the traits of interest. So this is like just trying to, we need to kind of ask those SNPs. So we ask SNP by SNP if they are just related to the trait. It's like trying to see if there are SNPs holding flags or holding signs saying the gene is here or the gene is around here. This is in a nutshell, just to keep the things very simple. And then when we use those statistical methods, this is what we see. So, and this we call Manhattan plot. So this is based on GWAS. So one of the speakers before talked about GWAS. So this is what we call GWAS, right? This statistical method. So this example here, this picture that I'm showing, it's not from sequence data, it's from regular 50K SNP chips, but it serves for the purpose because I just want to show how it works. So we have here each chromosome in the XX, sorry, each chromosome just and they are represented by different colors here, right? And then in the y-axis, we have the significance level. And in this case here, we have a Manhattan plot for birth weight in Angus. So we are trying to identify the SNPs that are related or SNPs that can give a lot of information about birth weight in Angus. So each SNP is represented by one dot here. So let's say that we declare that the significance level is like 5.9 in this case. So the SNPs that are above this line, this red line, are the ones that we're going to say, okay, those are the SNPs that give a lot of information about birth weight. So they are the causative SNPs. We can call them causative SNPs, or we can call them selected SNPs. But we can be less strict as well and just say, okay, let's declare the SNPs that are significant, the ones that are above this black line here, which is in 2.2. So in this case, we're going to see more SNPs that are related to the trait. But the thing is, when we try to use some methods to try to identify which SNPs have more effect on the trait, we have some issues here. And the effectiveness of those methods, they depend on several things. So one thing is the effective population size, which is the number of males and females that are breeding in the population. And it also depends on the number of animals that have sequence information and also phenotypes. So if we have large data, so large amounts of genotyped animals or sequenced animals and phenotypes, we are more confident in finding those causative SNPs. But if we have small data, it's much more difficult to find or to identify those causative SNPs. And also this process is more susceptible to errors. So this is the problem. And just to illustrate what I'm talking about, so this is an example here of a study that Ivan Pokrynik did when he was doing his PhD in Georgia, so up to last year. So in this case, we investigated the effect of population size. So how a population size would impact the ability of SNPs to find real genes. So here we have results just for one chromosome, and then in the y-axis we have SNP effect. So here we simulated 10 genes of equal effect. So we had simulated data. With simulated data is very easy because we can just say, okay, we have 10 genes or we have 100 genes. So in this case we had 10 genes, so represented here by the red dots, right? And then we used, for each chromosome, we used 500 SNPs to try to trace those genes. And then we can see here, for the second gene, we could see one SNP that did a pretty good job. So we could see almost one-to-one correspondence here. But in some cases, like in this case here, we could not see any SNP tracing the gene. So we could not identify this gene here. So this picture here shows us that trying to find genes may not be so easy as we may think it is, right? So if we had a successful search in this case, we should be able to see one green dot close to each red dot, but this unfortunately was not the case. And we can also see this data in a different way, in a slightly different way. So if we just average the SNP effects across all chromosomes, we can see the distribution. So we can see how the SNP effect is distributed across the genes. So in this case here, we had a population with effective size of 60, and we used 6,000 animals with genotypes and phenotypes. And we could see here one SNP that had an effect of about 2.8, but in this case, the gene effect was 4. So we could see it's almost there, right? And then we decided to genotype and phenotype more animals. So instead of 6,000, we had 18,000. And in this case, we had a better power to detect the gene, to try to find the gene. So because we can see here that there is one SNP that has an effect of about 3.5. And if we say, okay, the threshold to detect or to determine which SNPs are significant is 3, we may lose this SNP here. We are not going to select this one, but we are going to select this one in a larger population. So if we have a larger population, we may be able to do a better job trying to find which ones are the causative SNPs. And so we don't have just a problem with small data. So I showed you before that if we have small data, we don't have enough power to actually figure out or to discover which SNPs are truly causative. So this is very important, population size and also effective population size, as I showed you before. But the method that we use to try to identify those causative SNPs is also important. So here we had 3 Manhattan plots, and this study it was done by a PhD visitor from Italy that came to UGA. He stayed with us. He's still here, but he will stay with us for almost 1 year. So his name is Enrico Manchin. So in this case, he investigated the impact of methods. So methods to try to find causative SNPs. So the first one that he used was the classical GWAS. So in this classical GWAS, it does not account for population structure. And then we had here Emacs and single-step GWAS that account for population structure. And then here in red, we can see the simulated genes, so the genes that he simulated, and a stronger color means a larger effect. And if we use the classical GWAS, here, just another point here, this blue line is the line to determine which SNPs are significant, so which SNPs are affecting the trait. So here we can see a lot of SNPs that were declared significant, although some of them or most of them were not even closer to SNPs. And this was using also a small population. But then when we used Emacs or single-step GWAS, we could see here that some genes were correctly traced by SNPs, but still some genes were not able. So we were not able to find some of the genes. So again, for the second time, this shows us that trying to find genes may not be so easy, right? So by knowing that trying to find genes may not be so easy, now I just want to shift to some results using real data. So we have some results on beef cattle and dairy cattle when we selected SNPs from sequence data and used those SNPs to try to see how much increasing accuracy we could get. So the first example that I'm going to show you is a study done by my PhD student, Sambong Yang. So Sambong did this study, or the main portion of this study when he was doing his master's back in South Korea. So he used data from Hanu beef cattle. So Hanu can be compared to Wagyu because it has a very high marbling. So in this case here, Sambong used data from over 500,000 marbling score records. In the pedigree, there are about 1.3 million animals. And from those animals, 1,160 were genotyped. So they just had regular SNP chips, or they had 50,000 SNPs or 777,000 SNPs. And then some of those animals were sequenced, and then they were imputed to sequence. And so all those 1,160 animals were imputed to 11 million SNPs. And then after the imputation, they used some statistical methods to try to identify those causative SNPs, and they could identify about 331,000 SNPs. And then they used that for genomic predictions. So they use just regular SNP chip, and they also use those selected sequence SNPs or the causative SNPs in the evaluation. And then here is what he got. So we have results from base R, GBLUP, and also from single-step GBLUP. So the first result here in blue, we have accuracies when they used 50,000 SNPs, just the regular SNP chip. And then in red, we have when they used the selected SNPs from sequence data. And we could see here that the accuracy was even lower than before, than using just the regular SNP chips. And then in green, when they combined the 50,000 SNP chip and also the selected SNP. So we could see that the accuracy was restored to the same level as before with the 50,000 SNP chip. But still, in this case, we did not see a gain in accuracy. And why is that? Well, there are some reasons here. First, this data is small. What I mean by small is that we have a small number of animals with sequence information, just like less than 1,200. And also in this case, those animals were used to try to select the causative variant. So we had a small set to select the causative variants. And because of that, possibly those variants that we are calling causative, they are not actually causative, right? And then if you are wondering why the accuracies from base R and GBLUP are lower than single step, and this is basically because in the first two methods, they could just use 1,200 records, but in single step, they could use all records available. So another example that I want to show here is with a larger data set. So this study was not done at UGA, so it was done by Paul van Reyden from USDA. So they had sequence information for about 27,000 US hosting bulls. So some of them were sequenced, and then this information was imputed to 27,000. And then based on that, they used some statistical methods to select about 16,000 SNPs based on effect size of 33 traits. So they selected the SNPs that had higher effect on about 33 traits. And then what they did, they compared just like using the 60,000 SNP chip and adding those 16,000 selected SNPs to the 60,000 SNP chip. And then what I'm showing here is just the gain in accuracy for 33 traits and also for the average. And here, what I did, I just got the results from their paper. So I just got 60,000 plus the selected SNPs minus the accuracy for 60,000 SNP chips. So if the difference here is positive, it means that there was a gain in accuracy. And if it's negative, it means there is a loss in accuracy. But overall here, we could see that the gain in accuracy was there. So there was a gain in accuracy by using those selected SNPs from sequence data. But this gain was small. So the gain was a little bit over 0.01 on average. And for some traits, this gain was a little bit less than that. So for milk, fat, and protein, we saw a gain of less than 0.01 in accuracy. But for some traits, it was a little bit higher than that. So for stature, we saw a gain of almost 0.03. And when Paul did this study, he used a method called base A. That does not consider all the information in the system. So we wanted to test single step with this exactly the same data. So we asked Paul to send us the data. He sent the data, and then Breno Fragomeni, who is he was a postdoc in our lab, he ran this study. So in this study here, we then tested again, so how single step would perform when we use 60,000 SNPs and 60,000 SNPs plus the selected variants, and this is in green. So when we use selected plus SNPs, I'm showing here in green. And then, so okay, when Paul did his study by using base A, there was a small gain lower than 0.03. And when we did this study at UGA, we did not see an increase in accuracy. And then the question becomes, so why do we have a small increase in accuracy, or most of the times we don't have increasing accuracy? So why is that? Well, in fact, we don't know if those variants that we are calling causative variants are actually causative. And in real data, it's hard for us to figure out if those variants are really causative. It's almost like impossible, I would say, if we want to know all the causative variants. But when we have simulated data, we can simulate the genes and we can see how the things look like. So Breno also did some simulation studies. So he had about 30,000 genotyped animals. The animals were genotyped for 60,000 SNPs. And then we assumed that this trait we were simulating was affected by 100 genes. And I'm calling here the genes also QTNs, or quantitative trait nucleotide. And then what we did genomic predictions using single step, only with SNPs plus the genes, and only with genes. And this is the accuracies that we got. So first accuracy is when we use just 60,000 SNPs, just the regular SNP chip, so 0.49. And then when we added here the 100 genes to the SNP set, but we did not give any information about the genes. Actually, we did not tell the evaluation system that those were genes, actually. So when we did that, we saw an increasing accuracy. So accuracy jumped from 0.49 to 0.53, so a gain of 0.04. So, and this gain was very similar to the gain that we saw using real data, right? But then when we used those 60,000 SNPs and we added 100 QTNs and we told the system, okay, we know the proportion of variance that each gene is explaining. So we put the genes over there and we put the amount of variance they were explaining. And then in this case, we got accuracy of 0.99. So why we did not get accuracy of 0.99? So this was 0.89. Why we did not get accuracy of 0.99? Well, this is because we had SNPs and we had genes. So we had to split the information from the genes with the SNPs as well. They were kind of stealing some information. And then when we use just the 100 genes to run our genomic evaluation, we saw an accuracy of 0.99. So this is perfect accuracy, right? But the thing here is the assumption that we made was pretty strong. So we assumed that we knew all the genes. But in fact, we don't know all the genes. So let's say that we assume that we know just 10% of the genes. So in this case, we are going to see an increasing accuracy, but we are not going to see a full increase. We're just going to see a percentage of this increase. And another thing that we did in this study as well, Another thing that we did in this study as well, we said, okay, so when we have 60,000 SNPs plus the 100 genes, so let's put the weight for the gene. And then when we did that, the accuracy was maximized. And then we said, okay, instead of putting the weight or the variance for this gene, let's move the weight a little bit further, just pretending we didn't know the exact position of the gene here. So when we did that, when we moved the variance or the weight to the first neighbor, second neighbor, up to neighbor 27, we saw here that accuracy decreased. So what does it tell us? Well, it tells us that if we want to maximize accuracy of genomic predictions, we need to know all the genes, we need to know all the variants that they explain, and we need to know the exact position for those genes, for all of them. And then usually when I show this slide, I always joke that I will be glad to come back in 2050 to discuss more about those genes, to talk a little bit more about those genes. But recently I figured out that it may take 100 years or 200 years for us to have information about all or the most or like a great proportion of the genes affecting traits of interest. So based on everything that I showed you here today, the final question of my presentation is, so is it really possible to increase accuracy of genomic EPDs with sequence data in real cattle populations? Well, I have several answers for that. One of the answers is, okay, it's possible, and it's possible if we get to know SNPs closely related to genes, or if we know SNPs that turn genes on and off, or also if we know the amount of information that those genes are giving. The downside here is that we don't know all the genes. And also, if we know just a little bit of the genes, we are going to have just a little bit of increase in accuracy. So what I'm trying to tell here is that what we need to do, we need to try to find a way to squeeze the information out of sequence data in a way that this information that we get is non-redundant with the information that is already in the SNP chip. So what we just need to find a way to identify where is the information that is non-redundant to the one that is already in the SNP chip. And what I can tell you is that there are lots of studies right now on trying to, or a lot of projects trying to sequence animals and trying to understand if we are going to be able to get some information and if this information is going to be useful for us in genomic predictions. So my opinion about that, so why we have a lot of studies on that, this is because the animal breeding genetics community is always trying to find the best models, the best methods, and the best source of information that will help us to increase accuracy of EPDs. And because of that, it's going to help us to increase genetic gain. So the point here is we are working with research, right? So research may have positive outcome or may have negative income. So the thing or outcome, sorry, it may have positive outcome or negative outcome, right? But I think we always need to hope for the best, and we always need to try to exhaust all possibilities. So, and if we find something that is really useful, we adopt that. And if not, we are just going to move forward and try to see what else we can do to help us to increase accuracy of EPDs. So with that, I think I'm already out of time here. It's over 7. So I just would like to thank you for your attention and thank you for being here with me like so late. Mark?
Daniela, thank you so much. I think Mark's still on.
Yes, I am. Sorry,
Okay,
I was just trying to figure out how to get advanced to this last slide. So we do have a question, and I think it's for Daniela. It is, by using genomic testing and AI to select for a handful of economically important traits, do we run the risk of narrowing the genetic diversity of our cattle populations, leaving them more vulnerable to unanticipated environmental challenges, whether it be a disease or the effects of climate change?
What I see is that there's always have to be a balance. That's why we have selection indexes, right? So we put a little bit here, we put a little bit there. And what is interesting is that we are always going back and trying to see if we are losing something. And if we are losing, we try to correct for that. So in my opinion, I don't see that is going to happen very soon. So I think we have been able to try to manage that very well. And so if we estimate like variance components based on data like 15 years ago or now, we don't see a big change in variance. So this means that we are kind of doing a pretty reasonable job in trying to keep that. So this is just my opinion. Probably the previous speakers, they have something to tell about that as well.
Yeah, well, I guess I'll chime in. I think it's a risk when we have things such as, let's say, disease or environmental adaptability that we're not measuring and including in our selection indices. And I think the gist of the question is that that we reduce genetic variation by using genomic testing in concert with AI, and I think certainly that there is a risk of that. But the other side of that is, to avoid all that risk, we make no progress. And so I think it's better to make progress and accept the risk, and to always be looking out for what are the unanticipated consequences. And if we see them coming, then we may have to start measuring and including additional traits that we did not have on our radar before. What do you think about that, Danielle?
Yeah, so I think, sorry, I was reading another question here.
Oh.
Sorry, I didn't get that. I'm sorry.
That's fine.
I think if everyone has the same objective and we're all using AI, then we run that risk. If we have diverse objectives, and that's less likely to get us so narrow that we're not going to have any diversity.
Okay, and then we have another question. Besides the advantages of genomic information, e.g. generation interval and accuracy, A decreasing of additive genetic variation was also observed after genomic prediction implementation, which is not desirable in animal breeding. Am I right? If so, what are your thoughts on how to apply genomic selection aiming either achieving great genetic gain and controlling additive genetic variation? What would be a suggested approach to control that?
So I think this question is linked to the previous question, kind of, right? So
I think maybe so. I think you're right.
Yeah, I think it's kind of linked to the previous question, right? But there are always ways to try to balance that. As I said before, one way is to probably, because the genetic gain is not just based on selection, but there is the mating component as well. We can try to balance that with mating as well. And we can always try to see or try to investigate what's happening with genetic variants in this case. And then if we see some change, we go back and try to change something in the system.
Okay, and then another question is, having highlighted the disadvantages of SNPs, why don't we pursue only sequencing data, having been sifted for non-causal sequences, et cetera, assuming it's affordable?
Mm-hmm. So the way that I see that is, okay, if we think about like using all the sequence data, 30 million SNPs, I think there is kind of a computational, it's not limitation, but it's a challenge for us to do that. But if we think about just like selecting some SNPs and using those SNPs out of sequence data or sequencing, or just like looking at those SNPs, like that would be like less than 1 million SNPs or something like that, this would be feasible. And maybe Mark can comment on that based on what your experience is on Gencove, because we can see that we can have high imputation accuracy, right? So it's possible to have this information for like hundreds of thousands of animals, but it doesn't mean, as I told you before, doesn't mean that we're going to use the 30 million SNPs. We need to narrow down. And then I think based on that, we can move forward to evaluations. If we have like up to 1 million SNPs, probably we are not going to overwhelm the evaluation system. But then we need to see if this is really beneficial or not.
Yeah, so I think it comes back to the P much greater than N problem. That if we've got 30 million SNPs to sort through, if we have 100 million animals, we're maybe in pretty good shape. But if we've got less than 1 million animals, then it's going to be difficult, as Daniela showed, to identify what really are the causal SNPs, and we have to so we're not going to be able to get them identified all that accurately, but we can make improvements in prediction without knowing exactly which ones are the causal SNPs. And so it's a challenging problem, but I think it's one that we can make progress on.
Mm-hmm. Yeah, and then just to add to that and to reinforce that one very difficult task is to try to identify the SNPs that are associated with the trait. If we have like large data sets, large, like based on what Mark said, the NP problem. So in this case, we may have some problems with the methods that we have right now to try to identify those SNPs that are really giving extra information.
Yeah, so I think this kind of identifies what the challenge is going forward. You know, the low pass looks really promising in terms of being able to impute sequence in an efficient and cost-effective way. We haven't really had access to that level of information in general for so far. And so there's an awful lot that we're going to learn going forward.
Mm-hmm.
And is anyone else seeing questions? I believe that's all that I'm seeing.
Just one just showed up, Mark, in the chat box here.
Yeah, I just saw that. So the reduced size of the Brahmin population would be a challenge. What suggestions to bypass that and still improve the breed?
When you say the reduced size of the Brahmin population, actually I have no idea about the effective size in Brahmin. Do you have an idea, Mark?
My suspicion is that there's a fair bit of diversity there. I'm not sure that's exactly what was being asked. Maybe the question is that because it's a smaller breed, there would not be as many animals genotyped.
Mm-hmm.
And that is always a challenge when we're dealing with a smaller breed. And the hope would be that with sequence data, that we would be able to finally begin to share information between breeds, which has proven to be very challenging to do with the SNP chips. But the thought is that when we get to the level of sequence data, that it should be more effective to combine data across breeds, and that actually doing that may help to be another source of information to narrow down which variants are the most promising. But Brahmin is going to be the most difficult, or any Bos indicus is going to be a lot more difficult to try to share information from Bos taurus. It's just not going to work as well when we go across groups that are that distantly related.
Mm-hmm. So
within Bos taurus, I think there's some opportunity there. It's going to be more challenging with Brahmin. So, what suggestions to bypass that and still improve the breed? Keep doing what you're doing, or genotype an awful lot of animals.
And can I add something on that? Because based on my experience, we have seen that we have a considerable increase in accuracy of EPDs or genomic EPDs when we have more genotyped animals than the number of effective chromosome segments, which is dependent on the effective population size. So in this case, when we have more genotyped animals than the number of independent chromosome segments, we are better able to estimate SNP effects, so we can do that more accurately. So this is one of the limitations. So if we have few genotyped animals, we may see a small gain in accuracy. And then Mark pointed out, and this is one of the things that we are going to try to investigate in the pig sequence data project, is if we can find SNPs that are equally informative in different breeds, because we have several breeds that are genotyped in the pig data set. So we will try to see if we can find those SNPs. So if those SNPs, they are equally informative across breeds, we may be able to get SNPs from one breed and predict in another breed. So this may also help small breeds. So this is one of the hopes.
Mark, if I may, I think one other important point, particularly for a breed that may have fewer animals, is to take advantage of phenotypic recording density. So it becomes a bit wasteful if you have a lot of genotyped animals that don't have many phenotypes. So obviously all breeds, but particularly those that perhaps are smaller, need to ensure first that phenotypic data recording is as dense as possible, or I think the latter is moot.
Thank you for contributing that, Matt. That's an excellent point. Yeah, genotypes without phenotypes don't get us very far.
Yeah, genotypes cannot do anything without phenotypes.
And so then we have another question. Can't germplasm be conserved from past generations in the National Germplasm Conservancy to overcome the problem discussed in a previous question? And I think that's the one about the increase in inbreeding that comes along with genomic prediction and AI And I think certainly we shouldn't lose genomic resources, but we can get into an issue where a large portion of the industry population is fairly concentrated, and if it goes off in the wrong direction and we don't keep track of it and head off problems, then we do have some risk. Did you want to address that, Daniela?
Yeah, so I don't know, but my concern with that, so let's say that we have germplasm from previous generations, right? So, and then let's say that we are going to like create an animal based on that and introduce the animal in the population. So the genetic level may be a little bit below than what we are right now. So this is kind of my concern on that, but I may be totally wrong on that.
Yeah, it's a good, a very valid concern. The issue is that we can be, as the question brought up, we can be selecting for things that we think are making animals more productive. And if we're not paying attention, we can have unintended consequences from that. And one of the presentations yesterday pointed out that generally selecting for productivity has an adverse effect on general disease resistance. And we had a presentation earlier today on the brisket disease, and we don't really know whether the high elevation is the same as what's happening in the feedlot cattle. But it seems that there, what's happening is that the really high growth, really productive cattle that seem to be doing everything really well have maybe been selected for slightly smaller organ sizes. And then when we grow them out at 3,000 feet elevation, if everything goes well, everything works out well, but you put them under a little bit of stress and they get right to the end of the feeding period and they just overload their hearts and end up dying right at the end. And there have been examples of those sorts of things happening in other species as well. So I think that's the gist of the question. It's not something I lose a lot of sleep over, but I think it is a legitimate question. Any other thoughts on that? And did I miss any other questions? Well, seeing none, thank you so much, Daniela, for sharing that with us. We always appreciate your contributions. And at that, I think we'll turn it back over. Well, before I turn it back over to Bob, I want to be sure and thank Bob for all the work he's done in turning what was supposed to be a live meeting in Florida into a virtual conference and having very little time to get organized to do that. And I think Bob's done a great job with that. And I presume he may have some closing remarks for the conference.
state of Iowa and Iowa State University are proud to host the 2021 Beef Improvement Federation Annual Research Symposium and Convention. The convention will be located in downtown Des Moines with easy access to the airport, hotels, and local restaurants. Iowa State University is just north with its research and teaching farms. Join us in Iowa and experience how Iowa provides the beef industry with innovation to application.